Hello and welcome to Only Side No Fi. So, if you find this video helpful, do like, comment, share with your friends as soon as possible and subscribe too. So, in this video series, I'll be talking about immunological techniques, wherein in this particular video, I'll be talking about ELISA, which is enzyme linked immunosorbent assay. So, without wasting much time, let's go on to it. Starting with the introduction, this ELISA technique was originally described by these two scientists, and it is basically a plate assay wherein either 96 well plate or a 384 well plates are used, which are polystyrene, basically some kind of plastic plates which detects and quantifies. That is, it can both detect as well as quantify the amount of specific protein. Now, what I mean by specific protein is it can detect antibodies, antigens, peptides, proteins, hormones, etc. All these can be detected. Directly jumping to the basic principle of this placed, what this ELISA technique is about is first you coat the well plate, which is a plastic polystyrene plate, with antigens. So after this antigen is coated onto this well, um, some irrelevant protein is added to block the unwanted binding sites. Now, after washing this, a particular probe is added, which probably is an antibody. Wherein, so what happens is this antigens pre-coated on the well is attached to the antibodies or this antibody which is specific to the particular antigen. So antigen antibody reaction takes place. Either this is detected or a secondary antibody is added along with a signal molecule to detect the presence of antigens or antibodies. So the basic principle what underlies this ELISA technique is you add your sample antigens, then add a primary antibody. Either this primary antibody directly is detected or a secondary antibody specific to that primary antibody is added, which is detected. Now, there are different kinds of ELISA. So, before the kinds of ELISA, let me tell you about the signal detection. That is, these signal molecules can be either enzymed or fluorescent label. Fluorescent is a totally different kind of ELISA, wherein it is called as fluorescent ELISA or FELISA. So this signal molecule basically works as either the primary antibody is attached to an enzyme, that is via some biotin compounds or any other compound. And what it is done is when the enzyme is labeled with a primary antibody, substrates are added. So what happens is this enzyme converts the substrate into colorful product, which is detected using a spectrophotometer or even visually color change can be observed. What this technique does is if a particular antigen or antibody of our interest is present, obviously the remaining things will be washed off. So if there is presence of the protein of interest, there will be a visual color change. If there isn't, then there won't be any color change. Also, the quantification or the concentration can be obtained by a spectrophotometer, wherein the concentration of this colored product is directly proportional to the antigen of interest or protein of interest which we are interested in. So examples of enzymes which can be used for this labeling are horseradish peroxidase or alkaline phosphatase. There are many other, these are just two of it. Now jumping to the types of ELISA, wherein there are preliminarily four types of ELISA: direct, indirect, sandwich, and competitive. Talking about direct ELISA, wherein as I mentioned, antigen is coated on the well plate. Primary antibody, which is attached to the enzyme via certain molecules, is added to this antigen containing plate. And if our particular antigen is present, then the antibody will be attached and the remaining will be washed off. Now, this non-specific binding, because of this non-specific binding is prevented using first of all irrelevant protein and second is by washing step. So if a particular antigen is present, the substrate is added. Enzymes convert the substrate into colorful product and which is detected by a visual color change or spectrophotometrically. If our antigen of interest is not present, no color is detected. Next is indirect ELISA, wherein what first we did was primary antibodies were attached to the enzyme. In indirect ELISA, secondary antibodies are attached, wherein the antigen coated plate is added with primary antibody, and then another secondary antibody which is specific for the primary antibody FC region is added to the plate, and the remaining things are washed off. Now, and similar, further steps are very similar, wherein the enzyme converts the substrate into colorful product which is detected spectrophotometrically. Now, what is the use of this indirect ELISA is, first of all, it can detect presence of antibodies. Second is it is the secondary antibodies are used for signal amplification, wherein n number of more number of secondary antibodies can be attached to the primary antibodies, thereby amplifying the signal or amplifying the number of molecules which produces the color, and hence detection is easier. What sandwich ELISA does is, instead of coating the well with antigen, a capture antibody is coated to the well, which is specific for the particular antigen, and then the similar steps of indirect ELISA are performed. But what difference it makes here is, capture antibody recognizes an epitope which is different from the epitope recognized by primary antibody and on the antigen, so making it very, very specific for our particular antigen of interest. Next is competitive ELISA. This is very different from these three types of ELISA which I already mentioned. Here what is done is, because the name is given as competitive ELISA, of course there is some kind of competition between the substrates or the molecules to be targeted. What it does is, basically it measures the concentration of a particular protein of interest. So what is done is, first the antibodies are coated on the well. Next, what is done is, our target protein from the serum which is to be quantified is added along with a similar antigen which is labeled to an enzyme. Now what it does is, as you can see in the second this diagram, wherein first our if we have low levels of target protein in a sample, less number of antibodies will be occupied by this, and more number of our antigen labeled with enzyme will be attached to the added will be attached to the antibodies. Whereas if the sample containing the target protein is high, more number of target proteins will be attached to the primary antibody, and our antigen labeled enzyme will be less. So here you can see the competition between the R antigen which is labeled to enzyme and the antigen present in the sample. So what you observe is, if there is less number of target protein in a sample, less the sites are occupied and more the enzyme labeled antigens are attached, and more is the signal produced by adding the substrate in the particular mixture. So more signal is recorded. Whereas if the target protein is more in the sample, less binding sites are occupied by the enzyme labeled antigen and less signal is observed. And therefore you can, using certain graphs, you can estimate the or quantify the amount of antigen or target protein which is present in our sample. Now, there are of course other types of ELISA called as reverse ELISA, wherein we won't be talking about those kind of ELISAs. Now, coming to the applications, wherein first application is, Of course, ELISA is used for a broad range of applications. To name a few, it is used for clinical testing, wherein it detects early stages of cancers, wherein certain biomarkers are detected using this ELISA technique. Other is for infectious diseases, wherein antibodies produced during these diseases can be quantified, or amount of antigens can also be quantified. For food industry, it is used to detect the presence of allergens. Next is uh, a wide range of applications also include drug monitoring for transplantation, etc. It can be also used to detect viruses, wherein or virus titer. Also, it is used in cases of autoimmunity, wherein platelet antibodies can be quantified using ELISA. Of course, there are n number of applications. So, to list a few, these were a few antigens applications. Thank you.